Hunner type interstitial cystitis, HIC, is a chronic inflammatory disorder of the urinary bladder characterized by persistent pelvic bladder pain, urinary frequency, and urgency. Histologically, HIC bladder manifests subepithelial chronic inflammatory changes characterized by predominant lymphoplasmacytic infiltration, epithelial denudation, and pancystitis. Recent evidence demonstrated the immunological inflammatory nature of HIC, characterized by clonal expansion and increased CXCR3 receptor expression of infiltrating B cells, and upregulated expression of genes involved in immune responses and infection. Based on the histological evidence of epithelial denudation and immunological inflammation in HIC, we aim to develop a novel animal model for HIC via autoimmunity to the bladder urothelium in this study. A transgenic model uroOVA mouse that expresses the membrane form of the model antigen ovalbumin OVA on the bladder urothelium as a self-antigen was used in the study. Antigen OVA specific lymphocytes were generated by immunization of C57 black 6 mice with complete Front's adjuvant emulsified OVA protein subcutaneously. Splenocytes, a mixture of T and B cells of the OVA immunized C57 black 6 mice, were prepared 14 days after immunization and immediately injected to the uroOVA mice intravenously. ELISA analysis of interferon gamma levels in the culture supernatant was performed. Bladder histology, HE staining, and immunohistochemistry for a CD3 positive T lymphocytes and CD19 positive B lymphocytes. Gene expression levels of interferon gamma, TNF-alpha, and pre-substance P in the bladder tissues. Pain behavior measured by a von Frey anesthesiometer and voiding function by micturition cage were evaluated at each time point up to 28 days after adaptive transfer of OVA prime to splenocytes. Age-matched model normal uroOVA mice that were not underwent adaptive transfer served as controls. Each time point contained 3 to 5 mice for both groups. We observed increased interferon gamma production by the splenocytes, stimulated with OVA, indicated that the immunization induced OVA-specific immune cells in C57 black 6 mice. The inflamed bladders expressed significantly increased levels of mRNAs for interferon gamma days 7 to 21, TNF-alpha days 7 to 21, and pre-substance P days 7 to 14 compared with the controls. In histology, the cystitis uroOVA mice developed bladder inflammatory changes at days 7 to 28 with a peak at day 21 tested. The inflamed bladders exhibited remarkable mononuclear cell infiltration, increased vascularity, and mucosal hyperemia. IHC detection further revealed that infiltration mononuclear cells were predominantly composed of CD3 positive and CD19 positive B lymphocytes. Meanwhile, control mice showed no or few histological changes in the bladder over the course of all time points. These histological features reproduce the histological characteristics of human HIC with the first evidence of B cell infiltration in EAC models. The cystitis uroOVA mice exhibited significantly decreased sensory threshold of pelvic nociception at days 7 to 28. In addition, the cystitis uroOVA mice exhibited increased urinary frequency over the course of all time points with significant changes days 7 to 21 compared with control mice. In parallel, the cystitis-induced uroOVA mice also showed decreased mean body volume over the course of all time points with significant changes at days 14 to 28 compared with control mice. These results mimic the clinical features of human HIC. Adaptive immunity to the urothelial antigen induces bladder chronic inflammation characterized by predominant T and B cell infiltration, increased pelvic nociception, and voiding dysfunction. The present animal model reproduces histological and clinical correlates of human HIC, which could be a potential novel model for HIC.